Ever feel like transcription is one of those topics you kind of recognize but couldn't confidently explain under pressure? On the MCAT, that gap can cost you. In this video, we're breaking transcription down into a clear, step-by-step story from how RNA polymerase finds a promoter to how a finished RNA strand is processed and ready to go. By the end, you won't just memorize it, you'll actually understand it. And that's what gets you points on test day. Transcription is the process by which RNA polymerase synthesizes an RNA strand complementary to a DNA template strand. In molecular biology, this is the first step of gene expression, converting the information stored in DNA into a functional RNA molecule, usually mRNA in eukaryotes. RNA polymerase reads the DNA template strand in the 3 to 5 direction and builds RNA in the 5 to 3 direction, adding ribonucleotides that are complementary to the DNA bases, with uracil replacing thymine. A common MCAT trap is confusing the template strand with the coding strand. Remember, the coding strand matches the RNA sequence except for T versus U, while the template strand is the one actually read. Unlike DNA replication, transcription does not require a primer, which is a key distinction often tested. Another important detail is that only a specific segment of DNA is transcribed at a time, not the entire genome. This selective transcription is crucial for cell specialization and regulation. You should also connect this to translation, where the mRNA produced here will later be decoded into a protein. On the MCAT, you may be asked to determine RNA sequences from a given DNA template or identify directionality errors, so always double-check strand orientation and base pairing rules. Transcription initiation begins when RNA polymerase binds to a promoter region, often with the help of transcription factors. The promoter is a specific DNA sequence upstream of the gene that signals where transcription should start. And in eukaryotes, a well-known example is the TATA box, located about 25 base pairs upstream of the transcription start site. Transcription factors are proteins that help recruit RNA polymerase to the promoter and stabilize the complex, making initiation possible. A useful way to remember this is that transcription factors set the stage, and RNA polymerase is the performer that begins the show. In prokaryotes, a sigma factor helps RNA polymerase recognize the promoter, which is a difference the MCAT may test. Once bound, RNA polymerase unwinds a small portion of the DNA to create a transcription bubble. This is similar to helicase activity in replication, but is carried out by RNA polymerase itself. The enzyme then begins synthesizing RNA at the +1 site. A common MCAT question might ask how mutations in promoter regions affect gene expression. The answer is typically reduced or absent transcription because RNA polymerase cannot bind effectively. This concept also ties into gene regulation and epigenetics, where promoter accessibility can be altered. During elongation, RNA polymerase moves along the DNA template, adding ribonucleotides to extend the RNA strand. As the enzyme progresses, it continues to unwind the DNA ahead of it and rewinds it behind, maintaining a moving transcription bubble. Nucleotides are added one at a time to the 3 end of the growing RNA chain, forming phosphodiester bonds through a condensation reaction. An important feature is that transcription is highly processive, meaning RNA polymerase can synthesize long stretches of RNA without dissociating. However, it is also less accurate than DNA replication because RNA polymerase lacks extensive proofreading ability, though this is usually acceptable since RNA is temporary. On the MCAT, you may encounter questions about the speed and fidelity of transcription compared to replication, or about how mutations during transcription differ in impact. Another key detail is that only one of the 2 DNA strands is transcribed for a given gene, which reinforces the importance of strand specificity. This phase is also where regulatory sequences within the gene can influence the rate of transcription. connecting to broader topics like gene expression control and operons in prokaryotes. Transcription termination occurs when RNA polymerase encounters specific sequences that signal the end of transcription. In prokaryotes, termination can be row-dependent or row-independent. Row-independent termination involves the formation of a GC-rich hairpin loop in the RNA, followed by a series of uracils, which destabilizes the RNA-DNA hybrid and causes the polymerase to dissociate. Row-dependent termination requires a protein called Rho that moves along the RNA and separates it from the DNA template. In eukaryotes, termination is more complex and often involves cleavage of the RNA transcript followed by polyadenylation. A helpful way to think about termination is as a stop sign encoded in the DNA sequence that tells RNA polymerase to release the transcript. MCAT questions often test recognition of these mechanisms or ask what happens if termination fails, which can lead to abnormally long transcripts and disrupted gene expression. This concept also connects to RNA processing and stability, as proper termination is essential for producing functional RNA molecules that can be translated correctly. Eukaryotic mRNA undergoes post-transcriptional processing, including 5-capping, poly-A tail addition, and splicing, before translation. After transcription, the initial RNA transcript, called pre-mRNA, is not yet ready for translation. The 5-cap, a modified guanine nucleotide, is added to the beginning of the RNA and helps protect it from degradation and assists in ribosome binding during translation. At the 3' end, a poly-A tail consisting of many adenine nucleotides is added, which also increases stability and aids in export from the nucleus. Splicing removes non-coding regions called introns and joins coding regions called exons, producing a continuous coding sequence. A useful mnemonic is cap, tail, and cut to remember these steps. Alternative splicing allows a single gene to produce multiple protein variants, which is a high-yield concept for the MCAT and ties into protein diversity. Questions may ask you to identify mature mRNA sequences or predict the effects of splicing errors, such as exon skipping. This processing step is unique to eukaryotes and highlights a major difference from prokaryotic transcription, where translation can begin even before transcription is complete.